dream of microfluidics is perfect uniformity. Channels produce millions of droplets, each supposedly a carbon copy of the others. Engineers call them monodisperse, meaning same size, same composition, same conditions. It sounds like a recipe for precision science. Treat each droplet as a test tube, and the statistics become clean. But here lies the paradox. The more we try to make droplets identical, the more the faint fingerprints of their birth begin to matter. In many, isn't it strange that striving for sameness can actually magnify the role of difference? Every droplet has a history. When the interface snaps shut in a T-junction or flow focusing device, slight fluctuations in flow rate, surfactant distribution, or even tiny vibrations in the lab leave microscopic traces. Some droplets carry a faint skin of surfactant that formed a moment earlier. Others trap an invisible bubble of dissolved gas. Still others inherit chemical residues from the channel walls. These differences are nearly impossible to detect at first glance, yet downstream, they show up as variations in reaction rate, nucleation timing, or even cell growth. A monodisperse population becomes a choir where each singer has a slightly different timbre. Scientists who study droplets for chemical reactions or single-cell biology know this quietly. Identical recipes often give variable outcomes. One droplet crystallizes early, another resists. One cell divides twice, its neighbor only once. The mystery is not that droplets differ, that is inevitable, but that the very push for identical conditions makes hidden variability the key determinant. If all conditions were wildly different, noise would be expected. But when droplets are made uniform, even vanishingly small differences shine through, amplified by sensitive non-equilibrium processes inside. Some researchers now talk of droplet memory. A droplet carries the imprint of how it was formed long after it should have forgotten. Atomic force microscopy and high-resolution optics show subtle surface textures. Tracer particles reveal that flows during pinch-off can bias mixing for minutes afterward. These memories influence chemical yield and biological fate. Could it be that droplets, like people, are shaped as much by their past as by their present? The paradox of droplet memory reminds us that in complex systems, absolute sameness is an illusion. The more precisely we control, the more we reveal the ghosts of tiny variations. For microfluidics, this is both a challenge and an opportunity. Challenge because it complicates reproducibility. Opportunity because these memories may themselves become carriers of information. Perhaps tomorrow's droplet computers or chemical archives will deliberately encode history into droplets, turning what once was noise into a programmable feature. After all, what is memory but variability that persists? In microfluidics, one of the main goals is to create monodispersed droplets that are as identical as possible. Uniform droplet size enables reproducibility in assays, drug screening, and synthetic biology. Engineers tune flow rates, channel geometries, and surfactants to suppress variability. The assumption is that once droplet size is controlled, every droplet acts like a clone of the others. This ideal underpins much of lab-on-chip design and analysis. Yet beneath this uniformity lies hidden complexity. Droplet generators routinely produce emulsions with coefficient of variation, CV, in diameter below 2%. It is like stamping coins from the same die and assuming they are indistinguishable. Every droplet forms through a sequence of breakup events, influenced by fluctuations in flow and channel conditions. Tiny timing differences, local impurities, or even thermal noise alter the exact composition and surface state. These microscopic histories persist, embedding a memory that can affect later behavior. Two droplets of equal size may carry different chemical microenvironments. The drive for uniformity ironically highlights the importance of subtle initial conditions. Hidden diversity emerges in spite of mechanical precision. Droplets formed seconds apart can differ in surface protein adsorption depending on residual wall chemistry. It is like identical twins whose personalities diverge due to early life experiences. Surfactants, polymers, or proteins often stick to droplet interfaces during generation. Even trace coverage alters surface tension, permeability, and molecular partitioning. These differences accumulate into functional disparities between droplets. A single contaminant molecule can bias reaction rates or nucleation events. Thus, history at the interface writes unique signatures onto supposedly identical droplets. Surface scars are invisible, yet consequential. In droplet PCR, uneven absorption of primers at interfaces can skew amplification yield. It is like two glasses of pure water tasting different because one touched a lemon peel. Thermal and hydrodynamic noise cause slight shifts in breakup time, droplet velocity, or encapsulated content. Once created, these small differences persist because droplets are isolated microreactors. Instead of being averaged away, fluctuations freeze into permanent individuality. Each droplet becomes a time capsule of its noisy birth conditions. Ironically, striving for perfect reproducibility makes the residual noise stand out more clearly. Noise thus generates memory. A microfluidic stream may yield droplets of the same size, but with variable numbers of encapsulated cells. It is like shuffling a deck and dealing identical hands, except one card differs and changes the game. What looks like nuisance variability can in fact encode useful information. Subtle droplet-to-droplet differences can report on flow stability, contamination, or rare events. By analyzing these variations, one can infer hidden features of the microfluidic process. Variability becomes a diagnostic channel rather than mere error. Instead of erasing differences, science can learn to read them. The paradox is that imperfection reveals truth. Statistical analysis of droplet fluorescence variation can expose uneven reagent mixing upstream. It is like reading tree rings to reconstruct the climate in which the tree grew. Droplets used as microreactors often host biochemical reactions, which can amplify initial microscopic differences. Small variations in surface activity or solute concentration can cascade into divergent outcomes. The same recipe applied to many droplets yields heterogeneous products. This heterogeneity reflects the embedded memory of droplet formation. Uniform inputs thus generate diverse outputs. Microfluidic chemistry remembers its own past. Enzyme kinetics in droplets show broad distributions of activity despite nominally identical conditions. It is like baking cookies with the same dough, but each oven tray browns them slightly differently. When cells are encapsulated in droplets, droplet memory complicates interpretation. A cell's fate depends not only on its genetics, but also on the droplet's hidden history. Differences in nutrient partitioning, oxygen diffusion, or surface interactions bias growth. Apparent biological variability may partly reflect droplet individuality. The microenvironment becomes inseparable from the organism studied within it. Cell biology is filtered through droplet memory. Single-cell RNA sequencing in droplets shows biases linked to local droplet surfactant composition. It is like judging a child's performance without noticing the quality of their classroom. The paradox forces scientists to rethink what identical means at microscale. Achieving true indistinguishability may be physically impossible due to unavoidable fluctuations. Theoretical frameworks must include memory effects and initial condition sensitivity. Practically, engineers must design systems robust to droplet individuality. Rather than eliminating memory, the goal may be to control or exploit it. Science must embrace imperfection as part of the design space. New error-correcting droplet sorting algorithms are being developed to handle intrinsic variability. It is like accepting that no two snowflakes are identical, but still designing skis that work on all. The paradox is that microfluidics seeks absolute uniformity, yet every droplet is a repository of unique microscopic history. Striving for identical behavior only magnifies the role of hidden variability. At once nuisance and opportunity, droplet memory reshapes how scientists interpret results. What seems like error may instead be information in disguise. Identity at microscale is never absolute, only approximate. The challenge is learning to read memory rather than erase it. Surface nanobubbles persisting differently across identical droplets highlight history's invisible role. It is like trying to clone a song, but finding each performance carries a trace of the musician's breath.